seizure. And so about epilepsy, this is a disease that affects around 1% of the population over the world. It depends countries. And 20 to 40% of the patients with newly diagnosed epilepsy will become refractory to treatment. So one possibility for them is to undergo surgical resection of the focus where the seizure starts. And but it's not that successful, and only 50% of the surgery having good outcome. So one possibility is to try to optimize the outcome, and one direction is to use computational modeling with tools such as the virtual epileptic patient, but there is also many other initiatives over the world to try to identify better the focus and what kind of surgery can be done. And also computational modeling can help to try to identify some specific underlying causes of the that originates the seizure. And so one possibility is to go to personalized model of brain activity with a personalization to each type of epilepsy and or maybe for each patient. In this last case, usually we have clinical data at the whole brain scale, and so if we want to have information from the underlying mechanism, we need scale integration. So the way it is done today in large science in neuroscience and computational neuroscience is that we have different models and we can relate them depending on the dimension of the model and the scale it relates to. So we can go from detailed neuron model to point neuron models that will only keep the core dynamics, and then based on that we can build a large network model expecting neuron, and then we can have mean field approach to reduce to few equations and then connect them to go to large scale model, because it is computationally extremely demanding to build whole brain model based on detailed neuron models. There were some initiatives such as the Blue Brain project, but then we also end up with a very complex system, and it's sometimes really hard to extract and to understand things from such a big model. So today I will give a few examples of the relation between these different models. I don't know if you can see my pointer.
Yes, we can see it.
Okay, so I will give some example at a different scale from my own work. It's not. It's more general, and the idea is at the end to have a broader discussion on this scale integration questions. Because all the model we built are usually centered on a given scale, and all the other underlying mechanisms that may exist in a biophysical system. A biophysical system such as the brain are not taken into account because we can't go to atoms in such a model. So we use reduction. So there is a clear limitation, and then we to take into account the rest of the brain. Usually we consider it as the environment of our model, and it is through inputs, noise, or whatever other specific parameters that will represent what is not taken into account into the model. And when we go to a reduction, we usually reduced this part. We either cut, or I didn't represent here, or compressed this centered model, and with the idea that we will extend to larger scale. So the first model I present is at a single cell level. It's it is with the idea of building a point neuron model, reduced as possible, to get some biophysical interpretation and relation to pathological activity. So here is a model we built. It's a reduced extended Schke model. It is a 4-dimensional model with two fast equation that represents the membrane excitability and the gating variable. It is a Hodgkin-Huxley formalism, so there is these different currents here written in this form, and we consider the evolution of the concentration of ionic concentration on the intracellular space and the extracellular space. And so we have a slow subsystem that describe the evolution of this concentration. So we take into account this concentration evolution, and this lead to depending on the concentration of potassium in the external bath. The external bath correspond to the far distance to the membrane, so large baths, as we can find in slice electrophysiology. By augmenting this potassium concentration, we can see the emergence of spiking behavior and with oscillation in the nurse potential. Here is in red the sodium nurse potential and in green the potassium nurse potential. And this leads to this different pattern of spiking activity of the concentration of the potassium in the bath. And after a certain value, we see that it goes to pathological-like activities, such as this kind of bursting, seizure-like activity like this one with a small depolarization block, permanent sustained activity, and the permanent depolarization block, as we can find in spreading depression. What are the advantages of such a model is that as it is a reduced model, we can easily study the dynamics. And, so here is an example to understand how emerge this bursting behavior. So in dark blue is nucline of the V variable, and in light blue is gating variable N. And we can see that with the evolution of the slow variable, we have changed the shape of the nucline of V that will create a loss of stability in this fixed point here. And we can see the trajectory in red that will go to the limit cycle, where we have the spiking activity during the burst. And then we go through an homocline bifurcation and then go back to the lower fixed point here. So from such a reduced model, we can have this kind of separation of slow fast system. We can visualize and understand the structure of the underlying dynamics, but we can also characterize the type of pattern through onset and offset bifurcation. So this is interesting in terms of dynamics. However, it remains a reduced model, so it means that we remove many contributions to the dynamic to this this minimal model to get these dynamics. Here it's an example with external potassium concentration elevation, but we may find other model with other influence, other biological aspects that may lead to qualitatively similar patterns. So then I will give you another example at a spiking neural network. So now it's going from point neuron model to neuronal network. So it is in the other direction. It is not in the reduction, but it's going to the next scale. So here we built a phenomenological model of cortical dynamics based on the adaptive exponential integrated amplifier model proposed by Brett and Gerstner. And we built a very large network of 10,000 neurons with 80% excitatory in green and 20% of inhibitory cell in red, connected with conductance-based synapse. And we built a network like this that receive an external input. And from this network, here is a raster plot of the spectrum activity. We can measure the firing rate of each population. From such network, we can have some dynamics that correspond to healthy states, such as awake, synchronous irregular states, slow wave that correspond to the up and down states, and but we can also apply an incoming perturbation which correspond to seizure lag perturbation, which correspond to an elevation of the firing rate of the input. So this is here in blue. And in that case, we can observe two possible behavior of the network. Here here's the A and B are for the ADEX network. We can see that sometimes the firing rate of the excitatory population go to very high value, and sometimes the inhibitory activity may contain the excitatory activity. So in that case, if we consider a different region, that each of our model corresponds to a small region of cortex, in this situation, it may propagate to the next region, while here the seizure may stop to this region and not be propagate to the next one. So we also replicate this result with different other model with a slightly different phenomenology, but this is not the point here. Here it's for CADEX model, a conductance-based adaptive exponential integrated amplifier model, and here is with a point-norm model based on engineering slope. So we try to understand why we have this two behaviors. So for the different model, we apply different perturbation with a different slope. The slope corresponds to the onset of the perturbation and different amplitude of the perturbation. Here's the amplitude firing rate. And we find that the two behavior are possible. It strongly depends on the amplitude, but also slightly on the slope in that case. And what we see is that if we look more into detail, we see a cascade at the beginning of the seizure-like event in the propagating behavior. So the neurons are entrained into this paroxysmal activity. And so we try to understand a little bit better what happened in this situation and how we can study and maybe reverse potentially the two behavior and go it from propagating to non-propagating behavior. So to analyze such a large system, bifurcation analysis are not possible. We have a 4,000 equation. So we tried to other approach. So we've been interested in the defining some groups depending on the inhibitory, meaning how much inhibitory input each neuron is receiving from the network. So we define some subgroup, and we look into each subgroup as the mean membrane potential and at the variance. And we repeat this procedure for many simulations, and we find that there is a particular time window here where the variance is higher, and is into this window, if you applied a stimulus, positive or negative, you may switch the behavior between propagating to non-propagating behavior. So we did this, and interesting in this result, you can see this preprint in the archive where we discuss this more into detail. And this idea of dynamical shape of the incoming perturbation being crucial for seizure propagation is coherent with one possible taxonomy of seizure dynamotype, which has been published recently, with the idea that we can classify seizure depending on the onset and offset bifurcation types. And here, what we observe with this large dynamical system is coherent in that sense that it's going in the same way as a sharp incoming perturbation will more likely propagate than a slow one. We also identify a specific roles in inhibition, so this is interesting in this that sense. And but also the behavior can't be really predicted from a single realization. We have a 2 dimensional and 2 complex interaction, but it can be changed if we act in a specific time window. So this was an example where we have a 2 dimensional system to be able to understand it through tools such as bifurcation analysis. So one possible way is then to go to the reduction, to go to mesoscopic model, and then to go to large scale modeling. So here I will present rapidly some work on this possible reduction of the network model I presented before, and how we can go to large scale model. So we can define the mean field model. Here is one possible mean field model of ADEX neuron model that has been proposed. It's based on a formalism proposed by El Bustani and Destex 2009, and the version presented here is from Matteo Di Volo and Pao 2019. It takes into account adaptation as the ADEX neuron model, and it represents the firing rate activity of both population, inhibitory and excitatory, plus the adaptation curve. The variation. From this formalism, we can apply it to other type of model. It's based on the idea of transfer function here. That is based on single experiments and is specific to each neuron model. So we can derive this transfer function from a different type of neuron model, and then build the corresponding. We can build the corresponding mean field model. And here there is, if you're interested in this approach, there is this paper about this through different models. From this mean field model, we can compare it to the network activity, and in that case, that it is a much reduced model. We can study the bifurcation and do a stability analysis and so on. So here is a bifurcation diagram with here the external input, and here's the firing rate of the 2 populations. So in black are the fixed point of the mean field model, and green and red the steady state value of the corresponding spiking neural network. So we can see that it's follow quite well for low external input, and then then we lose little bit the accuracy, and then both goes to a very high fixed point. And this very high fixed point correspond to the one we observed in the spiking neural network during seizures. So maybe this fixed point, this very high fixed point in the Moonfield model can be considered as a model of seizure at this level. So this is to be studied further. And such a mean field model can be used in whole brain modeling. Then we can populate and build a network of mean field model. Here is an example of work done with human cryptography to build the network of mean field, and then we can observe the different dynamics at the open levels. So wake-like state and sleep-like state with AI and slow wave. And we do not have yet the simulation for epileptic seizure-like states.
Oh, perfect.
So conclusions. Yes, seizure can be modeled from single cell to whole brains. There is many models, and there is many much models than present here some few of my works. The model are centered to a specific scale, so this is important in the sense that often when people present work, computational work, the question at the end is, you do not take this into account, or you do not take this into account. No, we are centered on a specific question, so we are centered on a specific scale and level of description. So from complexity to dimensionally reduced model, it will depend on what kind of question you ask. Do you want to see this complex behavior, such as the cascade we see in the large spiking neural network, or are you interested in bifurcation? So this will give you different information of the same problem. So, but then you will need a different model. We can ask what we lost at each reduction step when we build Moonfield, for example, or when we build a point neuron model, how to integrate the scales. And maybe one way would be to do co-simulation, but then the interface between model is not trivial at all. And some points that have not been discussed here that seems essential is how we choose a stabilized scale, why a single neuron will be relevant, why region base will be relevant, and what is the size of the region, what is the information we need to take into account at the neuron scale, and then the degeneracy, because thousands of models may produce exactly the same dynamics, and why would be more relevant than another. And we also see this question previously, previous presentation. And finally, the hype for some model, because in neuroscience are made by human. We have big labs with famous people that produce a model, and they may spread this model more easily than other. And why one model or one direction will be better than another. And we also need to question the use of our model, and this strongly relates to the shift of the function of the model, because due to this fact, we can find many papers with models that are developed for a very specific purpose, that are too useful or relevant for specific questions, that are then used for completely different application, and may be out of the first assumption and maybe lead to wrong. So that's it for me. Thanks a lot. And,
thank you, Damien. Is there any question? Okay, Emre, please.
Hello, thank you, Damien, for the interesting talk. I have only one question. You mentioned this, not in the talk, but you mentioned that it's essential to understand how to choose the stabilized scales. But what do you refer to exactly when you say stabilized scales? It's in the last slide.
Yes.
In terms of spatial scale or temporal scale, or both of them?
Yeah, I think it is both of them. Go back to my first slide. The scale I'm referring to is what is here. We consider neurons cell because it's spatially relevant. It's bricks of biology. So we consider it as a stabilized, conceptually stabilized scale. It's not stabilized in terms of dynamic, but it's conceptually a stabilized relevant scale as a brick of it. So if for a single cell it's maybe relevant and we conceptually understand it, we also can understand that we can go to the membrane scale and maybe the channels, all the channels, the population of channel, and all the complexities that exist at the single cell level, which is already a complete world. But cell is one stabilized scale. And then when we go to spiking neural network or mesoscopic model or regional scale, it's not so easy to define. Good example of that is cortical column. There is no real size, well-defined volume of a cortical column, and also in terms of time. The time scale also is not easy how to relate what we measure, for example, in electrophysiology such as EEG or MEG, things like this, and what is really relevant into the dynamic of the brain.